Organization received first reports of a cluster of cases of pneumonia of unknown cause in China. The world has just crossed over 10 million infections and 500,000 deaths. In the words of the WHO Director General, the reality is this is not even close to being over. Although many countries have made some progress, globally the pandemic is actually speeding up. In Germany, there have been 193,000 confirmed cases of COVID-19 and nearly 9,000 deaths. The numbers in Australia have been comparatively much lower, with a total of 7,600 cases and 104 deaths. In both countries, the peak came in late March, with a significant slowdown in new cases since then. The Australian and German governments are supporting the global race for a vaccine, providing significant sums of new funding to vaccine initiatives, including CEPI and Gavi, and boosting domestic research and production capacities. We're seeing the results. A German-developed vaccine candidate is currently in clinical trials. German and Australian researchers are also making headway with vaccines in preclinical development. And Australian and German researchers from Griffith University, Fraunhofer, Helmholtz and Hanover Medical School are working together to develop drug treatments, but there's still a long way to go. Here to discuss global efforts to develop a vaccine, we're joined by two eminent scientists. Professor Peter Doherty is one of Australia's leading immunologists. He was awarded the Nobel Prize in 1996 for his discoveries into how the immune system recognises virus-infected cells. He's a Fellow of the Australian Academy of Science and patron of the eponymous Doherty Institute in Melbourne, which has been at the forefront of Australia's response to the pandemic and is currently investigating 4 possible COVID vaccines. Welcome, Professor Doherty.
Hi, good to be here.
We're also joined by Professor Marilyn Addo, one of Germany's preeminent infectious diseases specialists. Professor Addo is Head of Infectious Disease at the University Medical Center in Hamburg. With her work focusing on newly emerging infections, she's developed and tested vaccines for Ebola and MERS, and is currently also developing a vaccine for COVID-19. Welcome, Professor Addo.
Good morning and good evening to Australia.
Thank you. I want to begin our session today with talking about the origins of the virus. Peter, SARS-CoV-2, the virus that causes COVID-19, is thought to have emerged in bats. Millions of people around the world come into close contact with bats. Why haven't we seen a coronavirus pandemic of this scale before?
Well, that's a good question. We had 2 near events. In 2002, we had the original SARS virus, which came out of bats and infected people. Not nearly as infectious as this one, but it killed about 10% of the people it infected, much higher than the current death rates. And then, of course, we had the MERS virus, which came along probably out of the Middle East, but again from bats, maybe via camels. And that was a bit more infectious, as I recall, and also is still going and was killing about 30% of people, so a much more dangerous virus. And then suddenly we get this new pandemic virus. Now, I've been thinking about this, and I think this is the only truly novel respiratory virus pandemic in modern history, because influenza viruses, we've seen them before, and even though they may be novel in some senses, we do have cross-reactivities with immunity with other influenza viruses. So this is quite a benchmark, and I think we should be very much warned that these things are around and out there, and there are a lot more of them.
Yes, so to you, Marilyn, COVID-19 is another coronavirus, like the common cold. Why is this new virus so contagious and deadly?
Well, thanks, Paul. So, you know, we have been living with coronaviruses, and in Germany there are 4 circulating strains that, as you mentioned, cause the common cold and usually mild infections. But as Peter has mentioned, this is the 3rd big coronavirus pandemic potential that we've been facing in the span of less than 20 years. So 2002, 2003 SARS with the 10% mortality, and then the MERS in 2012 with like up to 40%, 30 to 40% mortality. But like we don't really know why SARS came and went. It just disappeared. So that's also why we couldn't really make a lot of headway there in terms of vaccine development. With MERS, it's a similar story. Like very few people, like globally, have been infected. So it's emerged in Saudi Arabia, and the big concern was actually at the time that having Mecca and Medina there, this respiratory virus could spread around the world, but in fact it isn't quite as contagious. So like those coronaviruses with high mortality are actually not as easily transmissible. And what makes SARS-CoV-2 so deadly in a sense is not its mortality rate. So we're working out the numbers still. So at the moment it's probably around 1%. It's more the fact that it is so contagious and it's spread quickly across the world, and we have asymptomatic carriers who can actually transmit the virus, and that is new, a new respiratory virus in a immune naive community, and this is a challenge that we hadn't foreseen.
Thank you. That's really interesting context from you both, just to set up now, I guess, the challenge for addressing this virus. Peter, historically it hasn't been possible to create safe and effective vaccines for human coronaviruses. Due to it being a new virus, there's also limited data on SARS-CoV-2. Given this, how likely is it that we'll be able to develop a vaccine?
I think it's pretty likely. I think Marilyn probably is more authoritative on this than I am. But really, the reason we haven't developed vaccines against the coronaviruses, the common cold coronaviruses, there are 4 of them, but there are a lot of other viruses, at least 100 viruses that cause the common cold. And we haven't really tried. And people did try with SARS, the original virus, but when it died out, the funding for that was dropped immediately. Kind of a pity in a way that we didn't go a bit further with it, but that was the reality of it. And it's also rather a pity we didn't go a lot further with antiviral drugs against the original SARS.
Well, Marilyn, if I could ask you as well then, how likely do you think it is that we'll develop a vaccine for COVID-19?
There are no guarantees, and like nobody can say, well, we'll definitely have a vaccine. There are many open questions. We have an unprecedented amount of vaccine research going on, and many consortia are on the way. Like there are over 200 vaccine constructs that are being developed in different stages of development. And we already have vaccines in clinical trials, and we know that the vaccines, some of the vaccines, at least in these initial trials, they're safe and they're immunogenic, so they do make T cells and they do make antibodies. So the big question is going to be, are those immune responses now protective against the disease? And we have like our track record with respiratory viruses, not stellar. So I guess, like the vaccine that has already saved many lives is the influenza vaccine, but compared to other vaccines that we know, it's not the best that we have. Let's just put it like that. But I'm optimistic, and maybe at this point, Georgia, we could probably, because people ask, why does it take so long to develop a vaccine? And so I'd like to put things into perspective a little bit. And Georgia, could you pull up that slide with the speed of vaccine development, just like an impulse for people on the screen? Let's see if that works. We have been. If not, it's also fine just to kind of mention it this way. So if you consider, and you had mentioned it in your introductory remarks, it's only been since December. This was the 6-month mark of like a new disease, and so it's only been in January that a new virus has been discovered. And the fact that we have had only 3 months between starting a clinical trial, the first vaccine going into a person's arm, and discovery of a new pathogen, this is absolutely record-breaking. We've never ever had something like that, and that is without kind of compromising safety or like the safety of the study participants. This is really outstanding. So I think those early vaccines will give us data throughout the summer and hopefully in the second half of the year some signals about protection or not.
Yeah, Peter, I wanted to ask you then, in terms of the efforts that are currently underway, are there any that are more likely to succeed than others?
That's a difficult question. I share Marilyn's opinion. I think it's highly likely we'll get a very good vaccine, maybe better than a flu vaccine. Flu is a strange beast. I mean, this is a strange beast too, but we'll see. And there are loads of viruses in development, of course, and Marilyn's probably more across that than I am, but the Jenner vaccine from the United Kingdom, from Oxford, is already well into phase 2 trials, maybe further than that. And I would be interested in seeing Marilyn's timeline or hearing what she thinks about when we will actually have a vaccine. My sense is that the Brits, for instance, have made a lot of that Jenner vaccine even before the final testing is done. They've taken a punt on it. But when you consider the expense of this pandemic, blowing a few $1 million away on making a vaccine is not that enormous. And so they could be ready to go later this year or early next year. Best estimates with the Australian primary candidate vaccine, which is the protein clamp vaccine out of the University of Queensland and Paul Young's group, is that it could be available mid-2021. It could be made in Australia, which is good because we don't have that much manufacturing capacity. Germany is in much better shape than we are in that respect. So I'm optimistic. I'm bullish about the vaccines, but anyone in science knows that we can be horribly wrong.
Thank you for your candour. Marilyn, how about from your perspective, can you give us some insight into the sorts of efforts that are being made to speed up what is still a long process and is important to meticulously?
So if you think back, so I think one time point that made a head start into where we are right now is actually the Ebola pandemic in 2014. At that point, the World Health Organization, or the world, realized that even for a not as contagious virus as Ebola virus, we were not well prepared. We had no medication, no therapy, and no vaccines. And so in the aftermath of that, the WHO convened a panel, an expert panel, and that panel was asked, so tell us what are the 10 pathogens that we think can cause pandemics in the coming years. And there were Marburg and Ebola virus were on there, SARS and coronaviruses and MERS were on that list too. And then if you scroll down, that list is updated every year. It has disease X. And so technically, COVID for us is disease X, but at the same time, it's actually, thankfully, a virus that comes out of a family that we already know. And in the aftermath of that, because usually we have been running behind. It was an epidemic, and we're trying to develop vaccines and therapeutics. And so at that point, it was decided, well, we have to be more proactive and try to develop platforms and vaccines ahead of time. And so, and that's also how CEPI started. So CEPI has the Coalition for Epidemic Preparedness and Innovation. They have their mission is to develop vaccines before epidemics hit. So a lot of the consortia who are now in the race for a COVID vaccine were already in races for other vaccines, such as MERS and the first request for proposals. So such as us, we were funded by CEPI to develop a MERS vaccine for another coronavirus. And so those consortia, usually it's never, it's always about teamwork. So you have different partners who can provide different skills. And same with Oxford. They were already in the MERS vaccine development. And so they could use the same platform and instead of having a MERS antigen in there, they could adapt it to a SARS-2 antigen. And so now they're up and running. And so I think this process has sped up development quite dramatically. And then we have new tools. So number one, as I said, just from the first pneumonia to discovery of the antigen or the pathogen was only a matter of weeks, and the sequence was made available very promptly. So essentially in January, people could start constructing their vaccines. And we have technologies that are very easy and easy, but very quick to develop. So a lot of the front-runner vaccines are RNA or DNA vaccines. They can be generated synthetically, and so they go rapidly in development into GMP production. So I think there are several components that add to the speed of vaccine discovery as we see it. And like, I'm not sure if we should try again, Georgia. So like maybe those first two slides, let's see if we can pull them up, because those actually show the front runners in clinical development, just to give people a picture of where we stand, and also it puts into perspective the speed of vaccine development compared to what we've seen in the past. So do not have here we go. So this is unfortunately you can't see the reference. It's actually a reference. There you go. So I just want to give credit to the people who put this together. So this is really breaking record times to first in human trials. You see the different vaccines and how long it has taken to bring the vaccine from development into clinical trials. And you see right on the top there, COVID-19, and it's been a tiny amount. So people say, because people keep saying, it takes so long to develop a vaccine. And in fact, we've never been as fast as this. And so if you go to the next slide, it just gives the participants maybe an idea of the front runners. This is as of June 20th. And so those are the 15 vaccines in clinical development that are on their way with their NCT numbers. The top four are viral vector vaccines, and this includes also the one that Peter had mentioned, the Chadox from Oxford. Then there are the greens are RNA and DNA vaccines. So some of those vaccine trials actually take place in Germany. And then in the bottom you see those inactivated vaccines. And those are clearly the ones that probably will give us the fastest results as well as to immunogenicity, and also then probably in terms of protection. But the protection part is the hard one, because obviously in Australia and in Germany, infection rates are going down, and so to prove that a vaccine is effective and protects against disease, it's getting more difficult in those countries. And I just wanted to give you that little perspective here.
Yeah, I think we should leave that slide up, actually, Georgia, and thank you for sharing that information, Marilyn. I think it was such a compelling example to show the comparison of vaccine development with COVID-19 as compared to other viruses and diseases. So thank you so much for sharing that. Professor Doherty, your institute, the Doherty Institute, is investigating 2 protein-based and 2 viral vector vaccines. How are those approaches different, and where are they up to?
I couldn't really give you the particular details on that. I know that they're all very much in the early preclinical testing stage, if that far. We're only now establishing a capacity to do a lot of virus challenge. Our big CSIRO lab that has been testing some of the vaccines, including the Oxford vaccine, is using ferrets. They're pretty much overloaded, and we've been having to expand capacity. But the Australian vaccines, other than the University of Queensland one, which I believe is in preclinical testing in ferrets and hamsters in the Netherlands, and is expected to go into phase 1 human trial in July sometime. So we're at a very early stage. Some of them from the initial just vaccination type experiments in normal mice looked as though they're giving extremely good antibody responses, but they're way behind in the sense of the global seed.
Okay, very good. Thank you for that. Marilyn, so to you, in terms of the work that you and your colleagues have been doing on a vaccine, how is that going? Obviously, moving at really sort of record pace, and you spoke to the fact that a lot of the work that has been done prior to COVID-19 has helped set up the platform for the work that you're doing now. How are things progressing?
We use a platform. It's called the viral vector. It's called modified Vaccinia ankara. This virus, the viral vector, in its form without genetic modification, is actually a licensed vaccine in Germany for smallpox. So, but that same vector can be used to to vaccinate, and it has a lot of safety data already. So, and we'll probably come to that later on. We may actually have different vaccines for different groups, and so this vaccine vector has already been immunocompromised in children and pregnant women. So it has a good safety profile. We have developed it. I'm part of the German Center for Infectious Diseases Research. In German, it's CISTIF. And we have a unit there called Emerging Infections, and it's a team of researchers in Munich, in Marburg, and in Hamburg. And so I think of vaccine development as a relay. So the vaccine was constructed by Professor Sutter in Munich, who has built the vector and has tested the vector in small animal models, and it's now being produced for GMP production. That's oftentimes a bottleneck because it's A, expensive, and B, we have mentioned previously, production capacity is limited in our countries, and actually the company that we're working with is currently producing a bird vaccine for another trial, and so they have to shift slots to accommodate SARS-CoV-2 vaccine. So we're at the same time as the other groups are doing the animal work and the company is doing the GMP production, we are doing the big regulatory piece and preparing the clinical trial. So we're scheduled to start in September with the phase 1 trial for COVID. And so we just published back-to-back with the Oxford group our MERS data, so we can compare it side by side, how these two coronavirus vaccines will fare, and maybe can draw some interesting conclusions from that.
Peter, if a vaccine is successfully and safely developed, how difficult will it be to deliver? You know, we struggle to deliver standard childhood vaccines across the planet in an effective manner. How can we ensure to get as close to universal COVID-19 vaccination as possible?
Well, this is clearly an enormous challenge to do that. Many of these vaccines, at least 12 of them, are being developed under the auspices of CEPI, the Centre for Epidemic Preparedness Initiative, and that includes the University of Queensland vaccine. Now, all those vaccines are developed, they're not patented, and the understanding is that these will be made globally available at the cheapest possible cost. And I understand that most of the vaccine companies have signed on to the idea that they will provide vaccines at cost. But manufacturing, of course, is a tremendous issue, and some vaccines will be much harder to make than others in very large quantities. As Marilyn mentioned, some of the nucleic acid vaccines, the synthetic ones, if they work well, and they're really rather new, but there are a number of them in test, they could be made in very large quantities pretty fast, and if we hope they work. I'm not sure about MVA. It's a very familiar vaccine platform, as is the adenovirus ones. There's at least several adenovirus vaccine candidates in progress. So the protein one that's being made from University of Queensland is, fortunately, we have in the CSL company a very large production facility at Broadmeadows where we could actually make 100 million doses of this vaccine a year, which is really impressive. But it's not just the vaccine itself and getting it out there. You also have to think, if you're going to vaccinate the whole of the population of the world, you would need at least 8 billion syringes. You would need 8 billion vials, unless you give it some other way, puff it up the nose or something, or give it in a patch. And if you needed 2 shots, well, you'd double those numbers. So the actual logistics of providing a vaccine like any of the vaccines globally would be massive. But there is an enormous commitment to doing this globally, and there's a lot of action. India, for instance, has traditionally, since HIV, made generic drugs, generic vaccines, and there have been agreements with the various companies that these will be made at low cost for use in the poorer countries of the world. That, for instance, the drug remdesivir made by Gilead is being provided and manufactured in India by a company exactly on that basis. So I think, in general, the world is working together very well on this. There is a commitment to global control, because I think there's also a great consciousness that this is a global problem, and if we're to return the world to normal economic activity, we have to solve this problem globally. It can't just be solved locally. So I think we're doing extremely well in that respect, except possibly for some of the United States leadership, but globally, World Health Organization's been doing a great job, and generally everyone's been coming to that party pretty well. But I'd be interested to hear Marilyn's comments on that, because she's much more directly involved in this.
Yeah, Marilyn, I did want to ask you as well, how has global collaboration assisted with the search for a vaccine? Any other reflections?
Exciting, because like these situations in the world can bring out the best of international collaboration. So we've just had this another Thunder event, and maybe somebody has watched on the weekend the artists that came together for the concert, and another really significant amount of money was raised for especially that cause. And there is no vaccine that is anywhere close to not even showing that they're working, and we have raised so much money to ensure that access is there, that we can enhance production capacity. A lot of risk has been taken to kind of buy contingencies of production capacity. It's really amazing, and I think the world has understood, and different stakeholders work toward the fact that we have to ensure equal access to this vaccine across the world. If we don't solve this problem as a global problem, then we will not succeed. We have seen how connected we are in this pandemic. So in a matter of weeks, now months, this virus has spread across the world. So a unilateral approach or national approach closing the borders is not going to tackle this. And we have to people say well, if we want to go back to normal, there might be a new normal. I think we have to talk about that as well. So it's not necessarily a given that we're going back to exactly where we were, and oftentimes it's said when the vaccine is there, everything is going to be back to normal. We have to be cautious about that and go stepwise. But I'm extremely enthusiastic about the amount of international collaboration that we have seen. And I've shown you how many consortia are already on the way. There is a lot of exchange. Some people say, well, it could be more exchange and more synergy, but especially with organizations like CEPI, they try to kind of share expertise so that not each of those consortia has to start from scratch. So there will be common standards, the new monitoring is standardized. So I think there are a lot of efforts being made to ensure that also the results that we're going to see are somewhat comparable. So if A does XYZ and another consortium does B, we have to also compare what these studies will show us. So yes, I think international collaboration is essential, and I really also applaud the WHO for their leadership in this, and CEPI as well. So and Gavi, the institutions who then also talk to the policy makers. So the scientists are trying their best, but I think this time I also sense that policy is already very early on in the game, making a big headway to ensure that we reach the common goal that we're set out to reach.
Yeah, let's just talk briefly about that new normal that we're living through at the moment, at least pre-vaccine. Peter, what if a vaccine can't be developed, or it could take some time from now? Could Australia, for instance, eliminate COVID-19 without a vaccine? And how viable is that kind of approach to other countries?
Well, if you'd asked a couple of weeks ago, we looked to be pretty close to eliminating, but of course we have a flare-up in cases in Victoria due to some breakdown with respect to security guards in the quarantine hotels. Eliminating it was never a strategy, but it's possible, but difficult, and it's not a possibility for most of the world. I mean, Australia and New Zealand, which have done rather well on this, both have the advantage of being island nations, and they also had the great advantage that we had very little community transmission before we woke up to it and started to limit ingress and so forth. Whereas in Europe and the United States, clearly the virus was in the community for quite a long time before it was actually detected in any serious way. So the ways out of this, I mean, one way out of it is a vaccine. The other ways out of it are if we could treat it much better. I mean, if we could ensure that people didn't die, or that most people didn't die, and we could make it the case that we could treat it and not nearly so many people got sick, then we could stop treating it like something super special. And if we had drugs to do that, for instance. And the development of antiviral drugs is extremely important. I mean, the other way of handling this, it's a very expensive way, but it reflects the AIDS model, that if a vaccine didn't work, and when it's very unlikely we'll actually get a vaccine for AIDS at this stage, then you handle it with antiviral drugs. And as we know, that's the way people live with HIV. They take a cocktail of drugs every day. But there's also another strategy, and that is what's called HIV PrEP. And what happens here is that people who put themselves at risk of catching HIV take a pill, which is two of the standard anti-AIDS drugs called Truvada, and they take that beforehand, and that protects them. So we could envisage that if we didn't get a vaccine, or if the vaccine did not work well in a key target group like the elderly, and that has been the experience with influenza vaccines, of course, then those people could take yet another pill. I mean, most of them are taking so many pills that if you shook them, they'd rattle anyway, so they wouldn't notice the difference. But that's a first world strategy, at least initially. Now, other possibilities are long-acting monoclonal antibodies, which are passive immunization that could be used as a protective. But if the vaccine's not protecting, I'd wonder whether they protect. And so these are the types of roads we could go.
Okay, that's fascinating. Marilyn, to you as well, treatments, what are starting to emerge as options in the short term while the search for a vaccine continues. What therapeutic options are most promising?
So, I do vaccine research, but I'm also the head of a clinical unit, and we've treated a lot of COVID patients, and so I think at the moment there are two candidates that emerged. Actually, we have a treatment working group and have a list of over 200 substances that are in clinical development or in considered for the treatment of COVID-19. Most of them at this point are repurposed drugs, and the front runner that Peter just already mentioned also, remdesivir, is actually a drug that was developed for the treatment of Ebola. And a couple of years back, there was a 4-arm trial in the context of Ebola, where 3 monoclonal antibodies and remdesivir were tested, and the 2 of the monoclonal antibodies did so much better that the remdesivir arm was stopped. Now this drug, has a broader activity, is being so it had already the advantage of having a lot of clinical data from these Ebola trials, and so it was already ready for human use, and it was applied very rapidly. This was also impressive how these trials could be set up so quickly across the world, and now we have a drug that actually has, in a placebo-controlled nice way, demonstrated that there is activity. I would say it's moderate activity. It's this is not going to be our saving drug, and so we do not have to develop any further treatments. But I think it's a very important first step to show, well, there is an impact that can be made with an antiviral drug. Part of the pathology in COVID-19 is also inflammation. So we see this second disease wave where people get really sick, lots of IL-6. There is data out there, which we have, I think, yet not seen the entire data, but there is a signal for dexamethasone that has actually the first benefit of mortality reduction as well in certain patient groups. And so we're looking forward to looking at the primary data on that. So that is actually 6 months into a pandemic. This is actually some progress, and now specific drugs are being developed. And I think while we're talking about the vaccine, it's critical that we continue also the development of other strategies and therapeutics, because it's going to be a while until we have delivered the vaccine to the world, if we have a safe and effective vaccine, and we'll probably still have infections such as we have with influenza, and we want to have tools at hand to treat those individuals as well. And I think Peter mentioned monoclonal antibodies as prevention, but I see potential there also as therapeutics, and those clinical trials will probably start in the second half of the year. There are plasma therapies on their way. The studies are not yet conclusive, but also in the case of Ebola, the recombinant plasma wasn't successful, while the antibodies, monoclonals, are actually treatments of choice now. So I do have some hope that we'll have another tool in our armamentarium against the disease with monoclonals as well.
Thank you so much for that. Certainly some reason for optimism. I thought I'd turn to some questions that have been sent in from our audience, and they continue to come in, so we will get to as many as we can. The past attempts to develop vaccines for SARS saw some adverse inflammatory effects when challenged by the virus. A rare but serious potential safety concern. Do we understand anything more about what causes this?
Should I take that? Paul, should I take this? So then, George, could I ask you to pull up the slides, because I think that's an important question to raise. It's the one with which says ADE. So it's actually, we always think that the worst case scenario is that a vaccine doesn't work, but in fact, the worst case scenario, this one exactly. The worst case scenario is actually if vaccines, like we usually give vaccines in healthy individuals. And so the worst case is actually the vaccine makes it worse, makes a disease worse. And we have actually seen that with some vaccines. For instance, an HIV vaccine trial had to be stopped because the people who were vaccinated had more infections than the control group. So this is a concern. We have to make sure that we address these things. And so for coronavirus vaccines, two potential challenges have been raised. And this is it doesn't say the reference. This is from an article from Barney Graham in Nature or Science. So if you want to look up the article, it's a very nice article, which kind of goes into this. And so two things are mentioned. One is antibody-dependent enhancement, ADE, and the other one is VAERD, like it doesn't really roll off the tongue, but vaccine-associated enhanced respiratory disease, which has been seen with other coronaviruses and other respiratory viruses. And I don't have to go into the details of how they are mediated, but there is an antibody-mediated part and a T-cell-mediated part, and has different mechanisms. All vaccine programs take this into account. We also try to address it in animal models, and CEPI has a working group on this actually. And at the moment, it's considered that the risk for COVID-19 vaccine development is considered low, but all of the consortia and clinical trials will address this and has specific measures in looking for any signal that could go in this direction. Because, as I mentioned, this is a critical piece that will make sure that this is not happening. It has been suggested in a non-human primate model, and there is a feline virus that has actually shown this quite dramatically, but this virus has a different tropism. So this is a very active, ongoing discussion in the scientific community, and it's being addressed. This publication is actually a good place to get some background on this.
Wonderful. Thank you for that, Marilyn. Peter, another question that's been sent into us, a range of factors such as genes and blood types seem to be linked to the severity of infection. Could we need personalised vaccines?
I don't think so. I think personalised medicines are kind of a thing we talk about in the future. There are some applications that depends on genomics and so forth. Personalised vaccines, I kind of doubt. I mean, Marilyn may have thought about this more than I have, but I think it could be the case that one of the reasons, say, that flu virus vaccines don't work so well in particularly some elderly people could have genetic basis, but I don't know any details. I think what Marilyn was saying is with these coronavirus vaccines, there has been this signal that came through from earlier attempts, particularly with the SARS vaccines. But as I understand it, everyone who studied those, who is now studying these new viruses and the new vaccines, hasn't seen anything like that, fortunately. But it is the reason why we have to go so carefully with rolling out this vaccine, because the situation is, as with Ebola, the only way you can really test the vaccine at this stage, after you've gone through animal testing and you've tested it for safety, is to vaccinate substantial numbers of people and put them out there in a situation where they can be infected with the virus. And we would tend to do that with younger people. And as younger people generally don't have such severe disease, we can't do anything that would put them at additional risk. So that's one reason why we have to go very, very carefully with these vaccines. And that will happen, there's no doubt about it.
Marilyn, to you then, another question that's been sent in to us is, how do vaccine approval processes work? Does each country have to conduct its own testing and grant its own approvals?
Yeah, that's a good question. And oftentimes regulatory aspects have been rate limiting steps. Like the first time that I've seen this kind of work very nicely in terms of not taking any shortcuts, really, as Peter said, that safety is of the utmost importance in this scenario. But oftentimes in regulatory processes, the whole approval process is consecutive. So you had to kind of gather some data, build a dossier, bring it to the regulators, and then only after that could you submit the next step. Now there are processes that are called rolling applications. You can actually, as your data, as your safety data, your stability data from the vaccines get off the press, you can assemble those, and the regulators will receive those. There's also international collaboration. So I know that our competent authority, which is in Germany, the Paul-Ehrlich Institute, speaks to the European authority and to the FDA there in very vivid exchange. Obviously, the regulators have different national laws, and sometimes they're not compatible. But in this international effort, we've seen like how fast it was able to go with the Ebola vaccine, and the speed here is actually even more rapid. The Ebola vaccine was had a conditional license approval by FDA and EMA last year in November, and which was a very, very short time frame for vaccine approval.
Very good. Yeah, thank you for that. Peter, question for you. What do we know about the variety of immune responses in people who've tested positive for COVID-19, and does the severity of symptoms have any impact?
That's really being worked through right now by a number of labs who are looking in very great detail at the T-cell and antibody responses. My understanding is there has been a paper saying that antibody responses can fall off very quickly. Not too clear about that. I think we're still waiting to see a lot of results. There can be some problem with T-cell responses. We're hearing that from a few labs. I think we have a paper that I'm supposed to look at tomorrow or the next day talking about this. But it's still early days. What's clear is, I think, that the majority of younger people who are either asymptomatic or not severely affected are probably immune to reinfection. There was some debate about that, but my personal sense is most of the situations where people thought they were discovering reinfection were probably persistence of viral genome, and they were picking it up with the very sensitive PCR test. But Marilyn's had a lot more experience than we've had in Australia. We really haven't had that many cases. So I'd be interested in her comments on that.
Yeah, Marilyn, I'd love to ask your response to that as well. And also just add on to that, are there greater clinical risks or side effects associated with faster vaccine development?
So I didn't quite get the second question that you asked. Can you repeat that?
So the second question is around, are there greater clinical risks or side effects associated with faster vaccine development?
Okay, so maybe coming back to natural immunity to COVID, we're also, so as Peter said, we have had a lot of individuals, and I think that the research, the data on T cells and antibody responses is emerging. I had to kind of look into this a little bit more because there was talk about a immunity passport in Germany, and quite really the data is not, the evidence is not quite ready for that yet. So what as Peter said, like in the vast majority of young immunocompetent individuals will between 2 and 3 weeks raise antibodies. We're looking at, obviously there's different classes of antibodies, and there's IgA, IgG, IgM, and depending on that, the seroconversion can occur earlier or later. Like some of the antibodies show cross-reactivity. As we mentioned at the very beginning, there are coronaviruses that have been circulating even before COVID, and there is a risk of cross-reactivity. So there are many things that we need to work out in our understanding of immune responses of natural infection. And as a matter of fact, now there are studies emerging showing that there are, like, yes, number one, that maybe antibodies wane quite quickly. And then we've talked about asymptomatic infection, and it has already been shown in MERS. If you have like a mild infection, maybe you are not exposed to as much antigen, and there are actually individuals, and there's a study from Germany, actually from Lübeck, that shows that is yet to be peer-reviewed. Like, so the nice thing is that we have all these preprint data now, that there are individuals with asymptomatic infection that don't actually raise antibody responses altogether. So that's why, like, I reviewed this data for this immunity passport, and we decided, or like my recommendation was, when this is not ready for prime time, we have yet to understand this better. The second question was speed and safety. That is my understanding. And like it's kind of interesting because on the one hand you think it's taking too long to develop a vaccine, on the other hand it's as fast as never, and so then safety concerns are raised. But that's, I think, we have to get across. We're giving these vaccines to healthy individuals, so there are no shortcuts taken that would put the safety of individuals at risk. So, like, we want to do something good with these interventions and not do harm, and that's why we go very careful, and that's why the perceived speed is slow, because we have to kind of really make sure that safety is ensured, and the side effects will be able to wait. Like, so far the data has come out there from the first vaccine candidate is very reassuring. So no severe adverse events, or like, at least to my read of the literature, and some of the platforms have already been tested in other scenarios. So the Chad vaccine has like a lot of good safety data from MERS, and that is also emerging for SARS-CoV-2 now.
Thank you for that, Marilyn. Peter, a question for you. Viruses are going to continue to evolve. What's the case for prioritising vaccination in research over treatments to reduce the severity of the virus? And could there be a point that we reach as we continue to investigate vaccines that there might be a shift, or do you think we're way off that and we need to pour as much energy into vaccine development as possible?
Basically, I think we need to pour, with this one, we need to pour as much effort as we can into every possible avenue, and that's in effect, I think, what's happening. I mean, as we've discussed, there are large numbers of vaccines being developed, but there are also other people with different specialties, particularly, say, structural biologists who are making designer drugs. The approach that's been used with HIV and the approach that we used to make the anti-influenza antivirals. And so they're pushing ahead as fast as possible. Other groups with that particular expertise have been pushing fast with the monoclonal antibodies. With a real emergency like this, that's causing so much economic and social damage, we just progress everything as fast as we can possibly go. I think the one thing that we didn't do that we could have ahead of time was really keep with developing antiviral drugs against the other coronavirus, because you can develop antiviral drugs that will work across a class of viruses. That's true for the influenza virus antivirals, and it's a pity we didn't really push on with the COVID. As it is, we've got Remdesivir, but that was developed for Ebola, and there are a number of other candidates under development now for the COVID. But I think one of the things we should do for the future, because CEPI has done the best you can do with pre-challenge or pre-knowing what the pathogen is vaccine development, because they were funding platform technologies where you can slot in some new viral genetic material and develop it. There are older platform technologies too, like MVA that Marilyn talked about. But you will always have a lag phase with a vaccine because you're doing something quite new if it's a new pandemic strain. With a drug, you can potentially develop drugs that will work across a whole spectrum. And of course, one of the reasons we don't worry so much about bacterial diseases when it comes to pandemics, except maybe with multi-drug resistant organisms like some of the TB strains, is because we have broad spectrum antibiotics. So if we can have somewhat similar, at least across a particular type of virus, antivirals, I think we'd be in much better shape. And I think we should be making them against any virus type that looks like a potential threat and could come out, say, of bat populations, which seem to be particularly threatening.
All right, well, we're coming towards the end of our time together. Just as a final thought then, Marilyn, to you first, what worries you about, the next phase of COVID-19 and what gives you hope?
So I think we've been busy trying to deal with the scientific and medicine questions, like primarily in the first 6 months of this disease. We're just now trying to appreciate the collateral damage, I say, economics and other. So I think that we will probably see in the months to come what effects that has done. But then on the other hand, while Australia and Germany have kind of curbed at least their first wave, and I think we were very well prepared for a second one. We see small little outbreaks, but I think we'll be able to handle them, and we will not go to capacity in our health systems, and I'm pretty confident about that. But seeing the disease of COVID-19 affecting Africa and India and other disadvantaged continents, worries me. And it's while the virus actually affects everybody, or it infects everybody, it affects, of course, different populations differently. And it's always Australia and Germany are in fortunate situations with democracies and stable health systems and a vibrant economy. And it's taken a hit on us, but we will reemerge. But it will affect other populations disproportionately, and that worries me. What gives me hope, and it has given me hope in the past, how the world can come together to face these really threatening situations together, scientifically as a community. I can think of small instances of neighborhood help in Germany, but also on the political scale. So that gives me hope, and that's why I also think that we'll be able to tackle this problem together. And hopefully in a couple of years' time, we'll look back to this webinar and say, we have a nice vaccine, and now we're on to our next virus. I'm sure there are going to be other situations like this, and we have to prepare for that as well.
And Peter, just briefly for you, what keeps you up at night and what inspires you, has inspired you through this crisis?
What's the question to me?
Oh, it's actually, we just need to unmute Peter, Georgia, and I'll give you a chance to answer again. There we go, Peter's off mute. To you.
Okay. There's still a lot we don't understand about COVID-19, and we're realizing that there are some very, very bad sequelae that are possible, and also very severe consequences, especially for people who've been on ventilation, that may be long-term. That's not uncommon with these severe virus infections, especially in elderly people. My sense of this is when we talk about 10 million cases worldwide, that has to be much, much higher than that, because many countries don't have the capacity to do a lot of PCR testing for background and asymptomatic infection. Whether it's twofold too low or it's tenfold too low, we really don't know, quite frankly. And a lot of the antibody tests, as Marilyn said, are still pretty suspect. The rapid screening type assays, they're not great. And so we don't really know background infection rates. But I think if we look at COVID-19 in retrospect, we say, well, this is terrible, but it wasn't the most terrible thing that could happen. But what really concerns me is there are much more terrible viruses out there, and there's no particular reason why we couldn't get a virus that's as infectious as this, but does kill very large numbers of people. It's not nearly as lethal as the 1918-19 influenza virus, for instance. So I think we need to rethink a lot of things through. I think this has been a kind of a trial run for humanity, because as long as we keep flying around the place in very large numbers, we're going to have this pandemic.
All right, well, Professor Edo and Professor Doherty, that's all we have time for today. Thank you very much for sharing your insights and expertise with us.
Well, it was great to be here. Thank you. Thanks to Australia and to Germany.
Yes, and thanks to everyone who has put aside time to join in this webinar today. There were more than 600 people who registered to attend, which is a great outcome. More information about today's webinar and future events will be sent to you in a follow-up email, and we look forward to staying in touch. If you'd like more information about the Australia-Germany Research Network and haven't joined already, please send an email. The information will be coming up on the screen. It's info.berlin@dfas.gov.au. You can also follow the network on social media. There is an active group on LinkedIn, or you can follow the Australian Embassy in Germany or the German Embassy in Canberra on other social media platforms as well. Thanks very much for joining us today for the webinar. I'm Paul Richards. Have a great day.